So as far as tissues go, is it still on? The 3 videos, so they're listed as parts to match the videos that are online. Again, you don't have to watch the videos online if you're in class, or they're as a resource. We're going to cover epithelial tissue today and the terminology, and then, like I said, hopefully get through most of connective tissue and then look at the histology slides. So this is the list of what you're going to need to know for the epithelial tissue, but the first item transcends all the tissues. There are 4 main tissue types. So you need to know the 4 main ones, and then this first part is going to go specifically into the epithelial tissue. So the 4 main tissue types are epithelial tissue that covers or lines. And every tissue in your body is going to be attributable to one of these groups. So this is kind of a big picture concept. There's epithelial tissue that's going to cover or line. So if we want to think in a general sense, because it's not really true, but anywhere a penny can go, you want to follow that. If you rub a penny on the surface of your skin, it's touching epithelial tissue. Well, so is the lining of your ear, and you can't stick a penny in there, but you know, you kind of get the idea. So you're running a penny on the surface of your arm, that is epithelial tissue. It's in your mouth. That's epithelial tissue. It goes down your esophagus. It's touching epithelial tissue. It plunks down in your stomach. It's touching epithelial tissue. And it continues the intestines, and it goes way back out the other side. And that is all epithelial tissue is your boundary between your outside world, which the penny would represent, and what is inside or what you're absorbing into your body. So I want you to expand your concept of what is inside our body versus outside our body. Being in the stomach, our body still thinks of it as outside. It hasn't chosen to absorb that yet. So it's still protecting itself by the epithelial tissue that's surrounding that's lining the stomach. And it's only in the intestines, if you break something down sufficiently, that we'll let it to be absorbed into the body. So think of epithelial tissue as lining cavities, lining the outside of our body. It covers or lines. Connective tissue does pretty much what it says, with a couple of exceptions. It connects things, so tendons, ligaments, fat's also considered connective tissue. Oddly enough, blood is considered connective tissue, and it's not really connecting anything, but it has the basic elements of connective tissue. So I want you to make sure we've got to categorize it somewhere, and it's sort of a loose term with regards to connective tissue, but it is in that family, as well as bone. Muscle tissue, we're going to talk about the 3 types of muscle: cardiac muscle, smooth muscle that's lining our vessels, lines our stomach, makes our stomach move food around, and then the one that most people are familiar with, your skeletal muscle. And then nervous tissue, that's what our brain and spinal cord and peripheral nerves are made up of. And the muscle and nervous tissue specifically, we're going to go into more detail in Unit 2 when we do muscle physiology and neurophysiology. So we're just going to generally go over the categories at this point here. So the bulk of this chapter for this unit will be on connective tissue and epithelial tissue. So epithelial tissue, the locations, there's many other places, but these are some of the general locations, like lining of the intestines. Like I said, anywhere a penny can go, it's going to line the intestines. It's going to actually form glands. Your sweat glands, your endocrine glands, they're actually made of epithelial tissue. And we're going to talk about the specific ones. Tubules. Kidney tubules are kind of like a gland because they're pumping and moving things around. So that's an epithelial tissue is used for that. The skin is our outer layer. It's used for protection, abrasion. That is made of epithelial tissue. So you can see it has a lot of different jobs. Even our lungs, the air sacs of our lungs are allowing for diffusion of oxygen to our blood. That is made of epithelial tissue. So we're going to go through the different types of epithelial tissue, and then you'll be able, when we're done with this, you'll be able to identify the shapes and locations and the functions of them. So the characteristics of this epithelial tissue is that the cells are jammed up really close together. And so I like to think of them as if you're at the Home Depot department and maybe you're going to be looking at tile. If you're going to lay tile, they're going to need to lay down on a basement membrane. So think of that as whatever the putty stuff is that you put down. It's got to anchor onto something. So all epithelial tissue has to have an anchor point. And so there's going to be different shapes, but they're going to be snugged up right against each other. Sometimes we might stack things. Sometimes we just like them to be one layer. So that's really what our epithelial tissue is about. It's going to have one free surface. If it's a tile, that might be the free surface of the room. And then the other surface is the basement membrane that anchors it down. So that's really the hallmark. None of the other tissue types have that. And epithelial tissue regenerates very well. So your skin, for instance, has a bottom layer that makes new cells on a regular basis. It's constantly making new cells because our outer skin layer is constantly, we're scratching it off, it's sloughing off. If we could look at each other like with microscopic vision, we would look like Pigpen from Peanuts, if you've ever seen that old school cartoon where it shows Peanuts walking along and it just shows like flies, it shows like dots all around him because he's like this stinky kid and stuff. And so that's kind of what we look like because our epithelial cells are popping off all over us. And so they're coming off because they're dead, so our entire surface is dead. We'll spend a little more time talking about why that is and the benefits of that when we get into the integumentary system in this. But it regenerates very well, which is why cancers like melanoma are some of the most dangerous cancers that we have. So you think it's skin, it's not like it's your brain, that's pretty important, you know, but the brain, although it would be a terrible place to have cancer, it is a much slower growing process than it would be on any epithelial tissue. And so that's one of the reasons why melanoma, once detected, can accelerate to be really, really bad very, very quickly. So because of their rapid regeneration. So this is where we get into some terminology. And so we want to make sure for epithelial tissue that we know that we have a first name and a last name. So its last name is epithelial tissue. So everything that you're going to write, you're going to say, like whatever it is, an epithelial tissue will be part of its name. Because especially on your tests, I'm going to need to know that you know which of the 4 tissue type big picture categories that we're talking about. And then the first name and the middle name, so we have the first and second name here, is it's going to be a layer. It's either one layer thick, that would be called simple. If you were to lay down, say, tile and there's like 3 of them stacked up, it will be known as stratified. And then the last one is pseudo-stratified, which obviously pseudo means fake. So it's fake stratified in that it appears visually to be stratified, but indeed it really is only one cell layer thick. So we'll show some examples here. So the first name just tells you what's the stacking situation. The second name is going to tell you what the shapes are, and it can come in 3 shapes. So I've been talking about tile. So tile is flat, is a pretty big square, but it's thin. So how many of you guys did in your prerequisite class, did the cheek, the skin swab of your cheeks, and you looked at them under a microscope? So those were all simple, or no, those were all the top layer of the squamous cells. So of the shapes, you have tile, which is going to be flat but thin. That's what you looked at when you did the cheek cell swab. So that's out, but it was actually the top layer of a stack layer, like a stack of pancakes. You end up swabbing that top layer of pancakes out, and that's what you're looking at. So if you were to look at it from straight on, this piece of paper, it looked like a big cell square or whatever shape you saw. But then if you had the opportunity, which you probably wouldn't on the microscopes, to see it from the side, it would be incredibly thin. So it would be like a piece of tile. And so that's known as a squamous. A cuboidal is going to be, you know, about as roughly, it's not geometry here, so it's biology, so it's like roughly as tall as it is wide, you know. Those guys are used mostly for pumping. You'll see them in tubules and glands and stuff. And then we have columnar. So the columnar are like bricks on end. They're going to be a lot longer. They're still going to be one cell layer thick, but they're longer, so they offer some protection. So these are the different choices that we have. So now let's put them together and actually make tissue types. Here's another picture just showing the same shape. So for instance, we see simple squamous, but this is stratified squamous. So it's still squamous cells. Now the catch here is the bottom ones look a little bit cuboidal-ish. We're just going to ignore that. It's just they're the plump, healthy ones. As a whole, it's going to be considered stratified squamous. In this case, the full name will be stratified squamous epithelial tissue. We have simple cuboidals, these little cobblestones, and then there can be cases where they're stacked up. Stratified cuboidal epithelial tissue. We have simple columnar, And then we have this pseudostratified columnar, because the pseudostratified ones, columnar, they're tall. What it is technically one layer in that each, at the bottom of each one of these cells is indeed touching the basement membrane, but some are short, some are tall, and the way the nuclei goes, it gives the appearance of it being stratified, when in actuality it really just is individual cells lined up next to each other. And then we have the last one, which I haven't mentioned, so I saved this one for the end, because it doesn't really fall into any organized category, is transitional. And that's only in one place of the body, and it's the bladder, because it could be stacked like stratified squamous at one point. That's when your bladder is empty. But if your bladder's full and it's stretched out, it kind of rearranges. So instead of being like 10 cell layers thick, and I'm just making up a number, it rearranges and now it's only 1 or 2 cell layers thick. It gets very thin. So it can transition, and it's the only place in the body is the bladder. It doesn't follow our little organized naming structure. Here we go again. We have the layers, simple, stratified, or pseudostratified. This is an example of a stratified. You can see how they're stacked up. This is an example of pseudostratified. In this case, pseudostratified is only going to be along the respiratory tract. So that kind of makes your life a little bit easier there. It's one layer of cells that appear to be more than one layer. So we can see, even though we got a little one here and a big one here, even this one looks like it's way up here, but it still has an anchor way down here. We have these other things are goblet cells, because this is the respiratory tract and you're coughing up mucus and stuff. That's where they come from. So they're not really counted in there. So pseudostratified columnar, because they're columnar, ciliated epithelial tissue is the name of that. This one, what do you think this one would be? Yeah, so this one's a simple columnar. So they're all roughly about taller than they would be wide. It's not going to be perfect. It's not like it's not geometry, but it's as good as biology can get. And so that one would be simple columnar. So we're trying to match these layers as well as shapes together. This is another way to sort of represent it, if you want to put it in the notes that way, where you can have a squamous, can either be simple or stratified, cuboidal can be simple or stratified. The columnars tend to be just simple. We won't be seeing any stratified, and there's a pseudostratified. So let's get into the actual naming. So we've, as you mentioned, simple, squamous, epithelial tissue. We have simple, so here's our first name, simple, and then we have the shape, and then we have the last name. So each time for the epithelial tissue, you need to include all of these. What is the stacking status? Is it simple or stratified or pseudostratified? And then the shape is going to be the middle name, and epithelial tissue is its last name. Of the simples, these are the ones that you're going to need to know. So I think it's often better to teach and learn this while we're looking at the histology slides or stuff I'll show either on the board or other images. The slides I have here are really so that you have the content, so you almost have a checklist to make sure that you know what you're supposed to know. But it's a little less fun to learn it this way. So I think, I'm hoping, yeah. So we'll go through each of these and then we'll show some examples in the body, because that's really how you're going to be tested on them. But all the ones that we're going to go through is simple squamous. What do you think that one's going to look like? Small, thin. And so it's going to be lining of our blood vessels and the alveoli. The alveoli are our air sacs in our lungs. So it is like super thin membrane that would be making the air pockets in our lungs, the alveoli. Simple cuboidals are going to be a little easier to identify. They're going to be a little like you're looking for one layer kind of in a row of little, squares kind of along. And then columnar, we saw that in a moment ago, they're going to be in the intestines. So when I'm just going to, while we have this here so we can try to make our notes more complete, simple squamous is ideal for diffusion. So the cell's not big enough to have much in the cell. So it's kind of so thin, like this piece of paper, that it's really meant to be kind of a barrier where there's air on one side and blood on the other side. So it's really meant to allow for diffusion across this thin layer. So that's really where you're going to see places like this. Cuboidal, it's in the glands, the kidney tubules. So you really see it, its job is pumping. it's really moving stuff from one side to another. You're going to either make sweat, or in the case of a kidney, you're going to say, I need to pump this into the waist, or you're just moving stuff around. So it's got to be thicker because it's got more work to do. It's got to have more space in the cell to get done what it's supposed to do. The simple columnar epithelial tissue kind of got a double job because it's in the intestines. And in the intestines, our job is to absorb the food that we ate. So we eat food and we digest it, and it's breaking down. So we want to be thin enough that we absorb stuff. So you think squamous. But squamous is so thin that there might be stuff that we ingested in, maybe the penny came in while we were eating our food, and it's got nasty stuff on it, and we don't really want to absorb that. So simple squamous would be too risky for our body to let harmful things crossing and into our blood. So we use simple columnar because not only do we want to absorb, so it does absorption, but we also want to protect. The intestines say, we need to absorb food. We need to absorb our amino acids and carbohydrates and things, but so we need to be one cell layer thick so we can absorb something. But you know what? Let's make it a really big cell, a columnar one, where we can actually give ourselves a little protection, keep some things out that we don't want to absorb, but then we're still only one cell layer thick so we can actually still bring stuff in. So for the purpose of the intestines, the simple one layer helps with the absorption, but the columnar helps with the protection element. So that's why it's ideal for that location. And then the pseudostratified columnar ciliated epithelial tissue is going to be along the respiratory tract, like your trachea and your bronchioles. And often ciliated, that's little hairs at the end, and the goblet cells. And we'll be talking about that. I'll probably mention it here. I usually talk about this with smoking when we talk about the respiratory system, usually in 202. But it's known as pseudostratified because you're bringing stuff in directly from the outside air, and so it's constantly growing. So you're gonna have tall ones and short ones, and so they're at various growing stages. And they're constantly trapping debris of what you breathed in. You have the cilia, that's little hairs at the end. The goblet cells make mucus. So you have kind of this snot goo stuff that's lining your trachea, so that as you're breathing in, if you're like, I don't know, imagine walking down a dirt road and it's all dusty, you're breathing in all that dust. We don't want it in our lungs, so that's hopefully sticking to the wall, sticking to the hairs, little cilia is bringing it back up, and you're coughing it up. So that's really its purpose, but our respiratory tract is getting exposed to a lot of harsh environmental things, so there's constantly growing new. So it's pseudostratified because it's at various late stages of development. So here we have the simple squamous. This is what it's going to look like. This is an example. If you're looking straight down the simple squamous, it looks like a tile, nice and big. But this here is a side view because it's nice and thin. So this is an air sac in the lungs, and this would be, say, looking down at the top of your cheek cells or the top of your skin. So this, your skin is actually stratified squamous. So it's like a giant stack of pancakes, but you're just looking at the top layer. This is one layer, but you're looking at it from the side. So that's simple squamous epithelial tissue. If we do one kidney tubule here, we have a little, sorry, I said columnar, simple cuboidal. Little cubes here. So this is our little simple cuboidal. So even in areas where you're not really seeing a defined line, you can kind of estimate that there's a line between these nuclei and be somewhat cuboidal, or somewhat as big as they are wide. So you'll see these in glands and kidney tubules because they are all about pumping stuff. Now our columnars, remember this is going to be in our intestines where it's one cell layer thick so we can absorb, but a big cell so we can keep some stuff out and protect ourselves. So this is part of our intestine. We have our little columnar cells like this, kind of along here. I'm not drawing all that well. So those would be our simple one layer of columnars. Stratified squamous, even though we're talking about squamous, yes, the bottom ones are a little more healthy. They're a little more cuboidal cells, but we're just not going to call it cuboidal. We're just going to call the whole thing stratified squamous from top to bottom. And this is the surface of our skin, and it goes from, so the whitish part is your dermis. So that's not what we're talking about here because the dermis is made of connective tissue. The stratified squamous epithelial tissue starts with the darker pink. So it has these up and downs because it's like a jigsaw puzzle. It's going to lock in with your skin. We'll talk about this when we get to this chapter about the skin. But all of these here stacked up. Stratified squamous epithelial tissue is from basically this layer all the way up here. So straight up and down. That's your stratified squamous. And because it's the skin, we have these funky different layers that you're going to learn the subgroupings here later. Okay, so of the simples, that's what we're looking at, what we just went through. Yes, question?
So could we think of stratified, the last one, as like mixed tissue, basically?
Just think of it only stratified. You're just going to see, because those cuboidals are a little bit bigger because they're the brand new born cells. And one of the benefits of it being stratified is by the time you get to the outer surface, it's flat and dead. And that's actually a really important component of our immune system, is that we're covered in dead cells. So that way, if someone sneezes on you, you're just like, that's dead cells, I don't care. You know, as long as you don't like breathe it in and like lick your fingers and, you know, you're like working hard to get it in your body, but it's kind of part of your protection. So yeah, they're born as cells, but then the further away they go, I thought it was on the note somewhere, I may not have mentioned it because I don't remember saying it. Epithelial tissue is known as avascular. And it's one word. I put a gap between the A and the V, but it's actually one word. Avascular means epithelial tissue has no blood vessels in it. So the blood vessels, for them to be born, has got to be down here in the dermis. And the further away it goes from where the blood vessels are, they're going to die off. And that's actually their purpose. One of their benefits is their death. Here's another example of, what do you think this little cell is here? These guys here. They're so thin that the widest part of the cell is where the nucleus actually pops up. So this would be simple squamous. And then you go for your epithelial tissue. That's the last name. Oh, I had the answer there, sorry. I never said that. I forget what's coming up. Okay, so we already know this one. This is a classic for the simple cuboidals. And looking at histology slides, that's why I want to give us a chance to do it today, as well as next Wednesday, is because it's like appreciating modern art. At first, you're just like, what is it? It's just like a mess, you know, and then you can start to like, okay, you know, interpret it your own way and actually start to see through some of these things. So at first, this looks like random circles. You don't know if you're supposed to look at this part or what's this going on here. But then for the purpose of what we're doing here, like this, we look at this slide when we do kidneys and we're talking about tubules, and I want the students to know about different tubules. For you guys, I just want you to spot the little squares and see if they're one layer in one of the tubules. So this is simple cuboidal epithelial cells. Again, we did this one. So I should have used different pictures for these. That's what I intended to do, sorry, when I was making these. This is pseudostratified columnar epithelial tissue. The cilia gives it away because it's always in the respiratory tract. We got short ones and tall ones, and it's still one cell layer thick. Okay, the stratified ones, those are the stacked up ones. So we're not going to do stratified columnar, so don't worry about it. You do need to know transitional. That's the one just in the bladder. So that one, just know that and associate transitional with the bladder and you're good. It's going to be super stacked when the bladder's empty and a lot thinner and stretched out when the bladder is full. Stratified cuboidal, just know it's more in glands, but we're not going to see them. So the only one that we're going to see all the simples we had on the other list, but we're also going to see stratified squamous, which I've already just shown you too. So those are things I want you to know about them and that they exist in the body, but we're not going to necessarily see them or you're going to be responsible for them on your test. So I've tried to cut back because there's a lot of slides that we're going to go through. So this is sort of my way of trying to not give you the whole shebang. So of the stratified, we have the transitional, obviously that's the bladder. And then the regular stratified squamous, we have this keratinized and non-keratinized. So for today, I don't care that you know the difference. I'm going to tell you anyways because I care when we do the skin. When we saw the stratified squamous, and I was showing you the skin, it was the pink slide, there was that dark line in the middle. That's the keratin, and that's in our skin. But if you have stratified squamous epithelial tissue, it's lining your esophagus too. So no different than if you're scratching your skin. Notice we can scratch our skin and blood isn't drawn because epithelial tissue is avascular. There's no blood vessels. If I were to scratch my skin so deep that I'm actually drawing blood, I've just gone past the epithelial tissue into the dermis itself. So imagine if you're eating, say we decide to all go to lunch together after here and we decide to go to a Mexican food restaurant and we have some tortilla chips. Maybe we're just really hungry and I eat one and I don't chew it as well and it's gonna be scratching down my esophagus. I'm gonna be pretty thankful that I have stratified squamous epithelial tissue that's gonna scratch down but not draw blood and still go on. So it's meant for protection, just abrasion, that kind of stuff. The keratinized is a specialty kind of feature added only to our skin. And it's kind of like our Gore-Tex layer, our own waterproof layer. And again, this is something we'll talk about when we talk about the skin. So this is more of a skin discussion. I just have it here so your notes are complete, especially when you're going back to study for the whole unit. But the importance is really about the waterproof layer of the skin. When we say waterproof, we're not preventing water from coming in. It's more of us from turning into a raisin. We're preventing water loss more than anything is helping to do that. So that's all the keratin job is to do. For the purpose of today, just know stratified squamous and what that means. So this here is a non-keratinized one. It's quite a beautiful slide of you've got your healthy bottom layer and you can see as it goes away from the blood source and from the basement membrane, how it becomes more nicely squamousified, made up the word, and it's stratified. So you can see how it goes there, but there's not that weird dark keratinized layer. So this is what you would see. So if you were to see it just like this as a picture on your lab practical for the exam coming up, then I would say, what type of tissue is this? And then you'd say stratified squamous epithelial tissue. If you just wrote stratified squamous, it would be half a point. So you have to include the last name. So I know that you know that the big picture family that it belongs to. And then I might say, where in the body would you find it? And then you could say, you know, even though this isn't keratinized, you could say, you know, lining of the mouth or lining of the esophagus and lining, you know, there's a few places that we would. So when you go back to the list where it has simple cuboidal, simple squamous, simple columnar, pseudostratified, know what the shape is and to name them, but know where in the body it would be. And I don't care that you identify where that particular slide is. So for instance, on the cuboidal, simple cuboidal cells, what was the kidney? If you were to say glands, that works for me. I don't care that you memorize that exact picture where it came from the body. I just want you to see it, be able to name it, but then just know anywhere in the body you could find that tissue type. Here's the non versus the keratinizer, both stratified squamous. We just have that extra layer, waterproofing. This is the transitional where it's empty bladder and full bladder because it's stretched out. So again, just know the transitional is associated with just the bladder. This is what it really looks like there, not the drawing. Okay, so as a summary here from the epithelial tissue is it could be, these are the places that they could be. And these are the jobs that it could do. So in the intestines, we're absorbing things. In the glands, we're secreting, pumping things. Kidney tubules also pumping. So these are some functions of epithelial tissue. The other thing I want you to know about glands is they could be endocrine, which means the gland is going to, the tissue is going to release a substance, but it only goes in the blood. So it's just staying in your body. Or it can be exocrine, and so I put the, in capital letters, there are differences here. The exocrine means it's going to go out a duct, say like a sweat gland. You have sweat beading on your forehead. It could also be because now we've expanded our view of what's in and versus out of our body. So anywhere a penny goes, if the penny is in our stomach, acid coming into our stomach is going to come from an exocrine gland because it's going out to that penny. That's still going out, at least in the perspective of our body. So exocrine glands will make a product, but it's going to go down a little duct and into some other space. Where endocrine glands, it's going to make a product from the gland, but it's just going to dump into the blood and then it's going to circulate. In the beginning of unit of 202, we start with the endocrine system, but you'd still have to differentiate those differences. So of the exocrine glands, that's going to be our primary focus. So there are two sudoriferous glands, and a sebaceous gland, which is whole. So we have 3 ways that these glands produce their product. We'll start with the easiest, which is going to be our merocrine or eccrine glands. You can call it either one. Merocrine, and it's a long, arduous name. Merocrine, sudoriferous gland, and then we have an apocrine sudoriferous gland. So the merocrine is the easiest one, and so we can see, and it produces it in this merocrine method. So this is where it gets confusing. We have a name of gland, and then we also have how they produce their stuff, their method of production, if you will. A merocrine or the eccrine gland does it in the merocrine method. And what it does is it's a cell, groups of cells, little cuboidal cells, and it's going to make sweat, just watery sweat, just like the beads on your forehead if you're nervous or hot. And so literally the sweat just kind of comes out. These little water droplets are going to accumulate at the bottom of that gland and then work their way out through the pores. And then you can see them beating out on your forehead. That's just going to be your straightforward, really simple merocrine, eccrine, sudoriferous gland. Then you have specialized places, and it's easier to think of animals in this sense, but we do too, because we like to think ourselves a little cleaner. But armpits, genitalia regions is where our apocrine glands are. So it's a little bit more than your watery sweat. So we have some protein chunks that get released out. In the animal world, those things are really attractive to other people, and maybe in the human world for some people, if they're not fermenting for a long time, you know. So the concept of pheromones comes really from these apocrine glands. So for instance, you know, like, anyway, you know, we're in farm country. Anybody smell like a billy goat? They're super stinky. That's their apocrine glands. We're at work, and a female goat thinks that's pretty good, you know. And so there's some actual physiological functions that are of that. For humans, it is the product coming out per se is not stinky. That's the person's own smell. It is the fact that there are fact that there's protein that are in there. It makes it attractive for bacteria to want to come to it and to consume it, and that's what the odor. Well, if someone has body odor and they're not showering very frequently, then that's where you're going to get it from. So it's not necessarily you're not inherently stinky, but you're actually providing food for nutrients for bacteria in your armpit region and stuff. So that's what that is. So there you can see in this image the cells actually producing some of the watery sweat, but also has the protein chunks with it. So that's going to be the apocrine component. And we are with the skin, again, on the skin lecture, we're going to be looking at the scalp, where we're going to have more of our merocrine, eccrine glands, because it's just going to be watery. And then we're going to have, and I think we have the palms as well. And then we have other regions of our body. We have some armpit, you know, ones where you can actually see some of the apocrine glands too. The last type is going to be a sebaceous gland, which is our oil glands. It keeps our skin supple. In fact, lanolin, like you look at some lotion, you're like, oh, it's some good lotion. It's got lanolin in it. It is oil from a sheep. So it's their oil coming out onto their skin. That's what's harvested in order to make lotion. So this sebaceous glands is what they're called, but it does it through the holocrine method. And the holocrine method is that the whole cell, that's why I always think of it a holocrine, except for it's spelled a little bit different, but the whole cell just gives itself up into the process of making it. So when oil is secreted, it's actually your whole cell coming in there, and then a brand new cell has to get made before the next batch goes. So that's another method. It also is why in those glands, those are associated with hair follicles, that especially people, teenagers, or so I'm talking about puberty, or you have women in their late 40s and 50s that's like puberty second time around, it's just the other way around, where there's varying hormone levels. And so what happens is, especially testosterone will increase production of these, but because there's chunks of cells and things, they can get bacteria invaded, and that's where you get acne. So then it's usually associated with those pores, but it's also with the oil. Because of the holocrine method, you have all of these extra chunks of cells in there, so it gives more for bacteria to want to feast on. And so that's often the root of the acne. Okay, so here's another depiction that you have this whole cell just ruptures. This one, just pieces of it goes. That's the apocrine, that's our pheromones, and then just your straight up watery sweat just sends out the water, but the cell stays intact. So you will need to know the 3 modes of secretion, the merocrine, apocrine, and holocrine, and an example of each. So the merocrine is going to be your watery sweat, your apocrine is going to be your armpit genitalia, more pheromone, chunky protein sweat, and the holocrine is going to be your oil glands, your sebaceous glands, and that's going to the whole cell ruptures. Now, epithelial tissue also includes hair and fingernails, oddly enough. This is the keratin side of things. We mentioned keratinization as our waterproof layer for our skin. It's a variation of that, actually. So the keratin that is our waterproof layer, our Gore-Tex layer of our skin, is formed in a little different way, but it makes our hair. And in a harder way, it makes our fingernails. If you're an animal, it makes the hoofs. And if you're a horse, those kind of things, that's made of keratin. A lot of it's also the horns of if you got a rope and steer in your backyard. That's also that. The hair, we're going to go through hair, is epithelial tissue, but we're going to see slides for hair, again with the skin, but we're not doing skin yet. And then the nails. So this here, although we're not going to look at them today because we'll see these when we do the hair, the skin slides with the scalp, see the follicles and the muscles of our hair. So this here is just so that your notes are complete when you're going through it. And so we'll be coming back to this. And the nails, these are the only 2 parts really that we have you know. So here's our fingernails, where obviously if you're free nail edge, however long you want to make that, that's your choice. That's just what's hanging over. But we have the main body of the nail bed. But what you can see here is the actual nail matrix is further back in your finger than where the cuticle is. And the whole point of the cuticle is to try to seal up and prevent any bacteria from getting under the lip here of nail folds. So you want to make sure the cuticles don't get damaged. So if people damage their cuticles too much, they get red and inflamed because you have a blood supply all through here, because this is where your new nail is formed from. And you can see where the bone is from that point. The lunula is this little half moon, kind of a white part. It's still pressed down as it's coming out from the nail root area. And so you don't really see the pink nature of your skin underneath it with the blood supply. It doesn't mean there's no blood supply there. It's just coming out so tightly from that, that radius point right past where the cuticle is. So that's why it has a little bit of a blanched color there. Here is visually what we're going to be looking at when we look at some of the glands. So we'll see these again when we do the skin slides. The sudoriferous glands are all over here. They're little cuboidals, and you don't even have to notice the cuboidal component. Just they are all these like little circles jumbled together. In actuality, those coiled tubular glands, if you can imagine, say, a garden hose, and if you sort of have it all strung out and then you decide, oh, I think I need to bunch it all up. And instead of putting it nice and coiled so it's all organized, say you just bring your garden hose and you're just heaping it into a giant garden hose pile. So the hoses are going every which way. That, and then take one hose and run it straight up. That's exactly what a sweat gland looks like in your skin. Jumbled set of tubes deep in your skin with one single part of the hose going out to the surface of the skin where the sweat's going to go out. So when we see a microscope slide of these, when we look at the histology slides, you have to remember how they're made. They'll take a section of skin and then they slice it like a guillotine, and they do this wafer thin slice. So think of the jumbled up garden hose that's every which way and then a single slice through it. You're just going to see a bunch of cut up holes at any given point. So that's why this thing looks like all these funky little holes. So you end up just seeing a whole gland will be clusters of these circles because you're just slicing through this garden hose. This here is our sebaceous oil glands. It's actually right adjacent to a hair follicle. The hair is coming out right through there. Bad picture, but you can see it's a good picture of the gland, not so much with the hair follicle. So what we should be comfortable with is the 4 main tissue types. Who can tell me the 4 main tissue types? Epithelial, we just did that one. Connective, muscular, and nervous. So I'm going to ask you that on the test, name the 4 main tissue types. And sometimes people we get so into the weeds, and I want you in the weeds sometimes too, you know, simple squamous, cuboidals, or all these like details. So oftentimes I think I'm asking an easy question, like, tell me the 4 main tissue types. This ought to be a gimme. This is an easy question. And then like half people get wrong because they'll be like, simple cuboidal, and like there's too far down in layers. So sometimes I do have questions that are like bigger picture that I want you to know overall kind of big picture concepts, as well as once we get into it, it'll be more smaller groups. So definitely expect that kind of question. What are the features and functions? When you think of epithelial tissue, tell me what's going to be unique about it. What is their job? Cover and line, exactly. So what are some features about it? If it's covering something and lining, it's going to have one side that's attached and one free open side. Is it absorb and protect? And where are we going to see absorb and protect? Simple columnar in the intestines. Perfect. Another one. Absorption and pumping. So we might do pumping like in glands or kidney tubules. Those are going to be cuboidals. Simple cuboidals. Transport is going to be the cuboidal. Or would we find a simple squamous epithelial cell? Alveoli and blood vessels. They're going to be our capillaries too. When blood gets the tissue and we want oxygen to get off the blood and into the tissue, that's going to be simple squamous right there too. The classification, so we just remember I have to say the layer, the shape, and then the last name. So that's pretty much that. It's there. Excuse me. So the types, we sort of covered that also. So you should know for each type on the list, where it's located in the body and what its job is. And if I were to show you a picture of, you know, simple columnar, then you can look at it and pretty much it'll only be the intestine, but, or a cuboidal. There's cuboidals in lots of different places. But even if I'm showing you simple cuboidal epithelial tissue in the kidney, and I were to say, where is it in the body? You say, it could be in the thyroid gland. Yes, it could be. So I don't want you being stressed out about memorizing the pictures of where exactly in the body they came from, because we do that when we do those specific units, whether we're doing salivary glands or whatnot. I just care that you can visually see it, and now you're training your eye to identify the shapes and what you're looking for and that it's epithelial tissue, and then just name anywhere it could be that it would be found. It doesn't have to be exactly what the picture is of. And you should know endocrine versus exocrine. Exocrine's going on a duct, endocrine stays in the body through the blood. And the gland types, merocrine, apocrine, holocrine. The merocrine is watery sweat, apocrine is like the stinky sweat, the proteins in it, and the holocrine is going to be your sebaceous glands, which will be oily stuff. And these we're going to go through the glands again when we get with the skin. Okay, any questions? Feeling good about that? Okay. So the connective tissue is a little bit harder. So, and then the other stuff's easier. So it's sort of like we're going up the hill here a little bit. The connective tissue is made of a group of cells. Obviously, we have lots of different kinds of cells. Because the connective tissue has the widest range, the connective tissue has the greatest range of kind of tissues. We go from blood to bone to like tendons, ligaments, fat. It has a lot of variety. But so there's going to be a number of different cells in the various connective tissues. The fibers in there also are going to be unique to where it might be. If you're looking at a tendon, it's going to be a lot of collagen. If you're going to be an ear, a lot more elastic fibers. If you're going to be in the spleen, you're going to have more of these reticular fibers. So you're just going to have fibers. So you basically have cells and fibers, the specific elements of which will be unique to different places, and this ambiguous ground substance. And this ground substance is really like the goo that holds it together, if you want to think of it that way. So you're going to have a cell and a bunch of fibers and a bunch of goo that holds it all together. And so then in this ground substance goo, it's actually where people take, you know, if you heard of like chondroitin sulfate or hyaluronic acid, people take some of these things from the health food store thinking they're helping their joints and whatnot. There are some of these components in the joints. The question is whether you take it from the health food store and you consume it, that it's actually going to make its way actually to where you think it's going to, rather than just buying it and digesting it and pooping it out. So, but that's the whole point of why people take those. Some of them can be better than others. And so really the basic element of connective tissue is you have cells, you have fibers, and you have some sort of medium that they exist within. And it's with this concept in mind is why blood is considered in this group. We have cells in our blood. It has fibers, although not circulating, but it can make fibers to make our clots. And then the medium that it's floating in is obviously the plasma. And so it's kind of a loose description of connective tissue, where if you're looking at, say, an intervertebral disc, you're gonna have some cells in there, some fibers, but the goo in there isn't gonna be watery like in the blood. It's gonna be a little bit more solidified, but it's still the medium that's staying there. So I really wanted to kind of have a general understanding of why connective tissue is classified as such. Here's the categories. This is where connective tissue gets a little bit more. We have these groups. We have a loose connective tissue, which really isn't designed for strength. Think of it as like packing material and other things that's going to be without our body that's not intended for strength, but more for form and structure. Then dense, that's going to be our strong stuff. Then we have cartilage, so that's going to prevent rubbing and protection. And then we have bone, that's actually just holding us up, and the blood. So these are all some of the categories. It's within each of these categories. Bone and blood are their own. Each one of these has about 3 subsets. So this is where connective tissue can get a little out of hand quickly unless you stay organized. Of the dense, these are the 3 you're going to need to know for the dense. There is dense regular, and it's regular because notice how everything's stacked up. Dense regular connective tissue is going to be aligned collagen. That means they're lined up in a nice, they're packaged nicely. So this actually is the strongest connective tissue type. So if you have a bunch of ropes, if we were to say attach some ropes to this side of the classroom and attach ropes to that side of the classroom and connect them in there, and you have a rope that goes across, and then we decide, you know, I'm worried that the walls are going to fall outward. Let me attach another rope. And by attaching more and more ropes in the same direction, you're going to maximize the force from point A to point B. They're not going to be very strong side to side, right? So this dense regular means you can pack a lot more in a confined space if they're packed up in the same direction. So not only do you maximize the strength because they're all aligned and all of the collagen is going from 2 points, point A to point B, but you can pack more into a smaller space because it's so organized. So that is the strongest, and this is what our tendons and ligaments are made out of. And so you think of a tendon just coming off your biceps brachii muscle, consolidating down, and then it has a single attachment point. That is going to be your dense regular connective tissue. And it's so full of collagen, that's the pink stuff, that the cell nucleus of the cell are these flattened black lines. Like they're just so full of collagen that it just mushes the nucleus off to the side and mushes them down because it's so packed full. That's why it's so strong. Dense irregular is our second strongest. As a second strongest, the fibers are jumbled in different directions because it is in the dermis of our skin. So when we pull our skin out, it's not just having to be strong from one point A to point B. It's not anchoring from one point to the other. So again, with the walls, we could have our ropes from one side to the other straight across. But if we wanted to put it dense irregular style, maybe we put an anchor there and go across the classroom that way. Maybe we go here. We crisscross it all over the place. That's going to be the collagen alignment in dense irregular. So it's not as strong because when you crisscross them, you can't fit as much collagen in, so it's not going to be quite as strong. And it has to be strong in multiple directions. So it's our second strongest, but it's still quite strong. In fact, I have this, and I for some reason has just found like the mother lode of my old Rock and Ice magazines. So when I was in graduate school, I was in grade and I liked to ice climb. So I would get this Rock and Ice magazine and I'm looking through it one day when I'm not working in the lab, of course. And in it was this commercial, and it was a commercial for carabiners. And because I'm a complete anatomy nerd, I see random everyday life things and I think of weird anatomy things about it. I'm looking through my Rock and Ice magazine one day, and on this page, I open it and I'm like, oh, that's an awful picture. And I look at it again and I'm like, oh, that's a really good example of like how strong your skin is, because I was teaching classes then too. So the picture is, so I have to describe it to you because I cannot find it, is this guy, he's a rock climber guy, and it's an advertisement for carabiners. And so this is the 1990s, so there was piercings and stuff wasn't as common, so it was sort of this shock impact ad. So there's this guy and he's leaning forward and he's got his nipples pierced with hoops. So all right, and the carabiners are linked to the hoops, okay? So there's that, but he's leaning forward, and so the hoops on the carabiners are clicked into chain, which is going to a cinder block hanging in. So he's got his hands behind him and he's leaning, and his skin is just like one, and so it's just kind of like, oh, that looks painful. But then it's a perfect example of seeing the skin stretched out, that you can see in this sort of conical nature of the pull that it's actually three-dimensionally strong, and that's what makes skin so strong. So the slide that you would see is all of this pink is like collagen mismatched in multiple directions, and that's what makes skin so strong. When you cut your skin, and we have to, seal it back up, and our body's busy trying to re-estimate the collagen, it seals it back up. You can kind of think of it like a sewing machine, and that's what a scar is. So you only have a scar because it's now organized, in that it's bringing the two sides together, and the organization of the collagen, rather than being just mismatched at all the directions, but it actually being more aligned, makes it more visually noticeable, and that's why you see scars in those cases. So this is multi-directional collagen fibers, three-dimensional strength. You're going to see it in the dermis of the skin. That's under our epithelial tissue. And then dense elastic. So it's going to be in your vocal cords. It's actually aligned. So we want them to be dense, lined in a similar way, but we don't want it to be, it's like a tendon. We want it to be a little bit movable. So that's where we have elastic tissue. So remember, we talked about cells and fibers and in some sort of medium. And so in this case, we just have the cells are densely packed or irregularly stacked, kind of messed up. Or we have collagen in these two, but this one's specific to elastin, elastic tissue, but it's elastin fibers. So they're going to be different fiber types. The loose fibers, or the loose connective tissue, we have just the easiest one is fat. So these are blood vessels. So that's not what the picture is supposed to show you. The fat is here. So let me circle. Here's the fat. Here's the fat. What adipose tissue looks like, so it's in the loose category. It actually looks like a cell, a nice white cell, and the nucleus is jammed over to the side. It looks like an eyeball to me from the side. And the nucleus is off to the side because the cell's main job is just to be filled with fat and full of lipids. So it's sort of like a semi-truck. The whole majority of it is going to be its cargo, and then just the front part's just the engine. So that's like where the nucleus is off to the side. So you can spot adipose tissue quite easily because it's like a lot of these are white circles that you'll see a little nucleus off to the edge. So there we go, fat. The purpose of fat is obviously for energy storage. So if we eat a lot of food one time, then it's our body's way to say, hey, let's save some for later. And so that's their energy for later, as well as protection. So it gives you, so we have fat tucked in different parts of our body to help pad and protect different organs and things. This here is a zoomed in view. This is actually where you can see the cells. So you can see here's the fat cell. Some of them, because remember the guillotine slice through it, might have missed the nucleus for this one, or you're getting a nucleus from another cell. So sometimes you don't always see the nucleus matched with the right cell. Then areolar, that's more light and airy. I always think of areolar connective tissue as packing material, sort of like the packing peanuts of our body. It's what's going to be surrounding organs. For instance, if you get like chicken meat from the butcher or from the supermarket and you peel the skin away off of the chicken and there's that weird stringy stuff. It's not really whole, it's not really that strong, you just peel it away, but it looks like spider webby stuff. That's this areolar connective tissue. Kind of holds stuff together, not that strong, kind of, that's used in packing materials around the kidney and different places. So it's considered to be our superficial fascia around the muscles, as well as surrounding some organs for protection. And this is what it looks like more close up. Kind of a mishmash of some collagen, some elastin. It's just kind of loose, a little more loosey-goosey. Reticular connective tissue, this one's pretty unique in one place, the spleen. And so what it's reticular, I want to think of that as like a spider web-like. Reticular is, and the spleen is just a weird organ. Picture a Ziploc bag, like a little plastic bag, and you stuff it full of cotton. There you go, that's your spleen. And you run blood into it, and if there's stuff that's got, ready to be snagged, like old worn out red blood cells, it's gonna get snagged on the cotton fibers, and then the blood's gonna come out. So it's sort of a way to filter stuff in your blood. And that's where the spleen, the reticular connective tissue, is this fibers. They're not meant to be strong. They're really like the cotton balls. So they're meant to just trap. There's like more of like a spider web-like formation to trap potential debris in our body. So it's meant for filtration, and you find it in the spleen. This is what it looks like close up. It's the black fibers, and then actually the cells around it are just the white blood cells ready to pick up anything that might be a bad guy in our body that have picked up any antigens. So hyaline cartilage is the most common cartilage throughout your body. For instance, if you can feel down your throat, your trachea, and it feels like if you run your fingers down a cardboard box, you can feel the ridges in that, just like you can feel the ridges down your trachea. That's made of hyaline cartilage. If you are at the butcher shop again and you get a bone, and the end of the bone, there's like this weird bluish white stuff, it's not cooked bone, then that's gonna be cartilage. Cartilage is gonna be capping the ends of our bones. That's what's gonna be in our joints. So if somebody has arthritis, it's the hyaline cartilage that's getting eroded away onto the bone. Fibrocartilage is gonna be other cartilage, but you can see there's more fibers in it. And then we have elastic cartilage, which is actually gonna be our ear. So back to hyaline cartilage, ends of bones, mid part of the nose. We want to think of our nose as having three parts. The way top part is bone, the middle part is hyaline cartilage, and then your bewitched end part of your nose is elastic cartilage. It's along the respiratory tree. It's the rings of your hyaline cartilage, the hyaline cartilage rings of your trachea, and you see it all over the body. Also has this glassy, so remember I said you have this goo that the cells and fibers live in. Well, there's really not a lot of fibers in the hyaline cartilage that we can see, but the goo that it lives in is a little more solidified, more like Jell-O, but more solid. Think of it as a glassy lake, and that's actually the name. Hyaline means glassy. Cartilage tissue, to me, visually has all of these like little hollowed out lacunae with the cells inside. A colleague of mine I used to teach with said they look like frog eyes to him. So he was always like, we'd go to the lab, does it have frog eyes? And it's cartilage. And then you could say, this is, the plain side is gonna be hyaline. If you got fibers through it, it's fibrocartilage. So fibrocartilage is only in two places of our body, or two, just like hyaline, except for we have more collagen fibers throughout it. And it's meant to present compression, not just capping the ends of bones like hyaline cartilage. It's actually meant to withstand a lot of weight pushing down. So it keeps it a lot stronger. So you're gonna see it in the intervertebral discs. So of our vertebrae, which we have right here. So between the vertebral bones, the intervertebral discs are made of fibrocartilage because we have so much weight coming down bearing on them. The other location that fibrocartilage is the menisci of our knee. So we have hyaline cartilage at the end of the femur and the tibia. And then this is what it looks like close up. You're still gonna get the frog eyes, but you have a lot of fibers. And then elastic cartilage is going to be more elastic fibers. You're gonna see at the very end of your nose or the ears. And then the last two, bone and blood. Bone is going to be its own chapter after skin. So we'll talk about bone in there. For now, I just want you to know bone and blood are in this connective tissue family. But we're going to get more detailed on bone later. In 202, you'll hear about blood. So as far as connective tissue, and then we'll go through and we're going to pare this down a little bit when we actually look at the slides. is what are the components of connective tissue? They're going to be cells, fibers, and ground substance. That's sort of the medium or the goo that it's going to live in. It could be really watery like blood, or it could be a lot more solid like bone or cartilage. Then we have what are some of the cells that form the matrix? Some of the cells are going to be osteo, meaning bone, sites. So this is where we're going to get some terminology. So you should know that osteo, obviously bone, and the end word for that site just means cell. You can have a chondrocyte, that would be cartilage. So a cartilage cell, a bone cell. The terminology that we're referring to here is if you have an osteoblast, it'll actually be one word, you're going to build bone. A chondroblast is the cell making cartilage. If you have a chondroclast, osteoclast or a chondroclast, it means it's going to break it down. Somebody with osteoporosis has very active osteoclasts, and you want to stimulate osteoblasts to build bone. But a normal healthy bone, and these will go into more detail when we actually do our bone chapter, but normal healthy bones constantly making new and tearing away old. So osteoclasts are a very healthy part of bone as long as it's well balanced with the osteoblasts. But there are definitely questions on the test that's going to say, what's the name of a cell that builds cartilage or builds bone, so that you're knowing the prefix as well as the differences between these suffixes. There was a question. Right.
Yeah, so when you break a bone and they say that it grows back together stronger, it like calcifies over, is that what's happening?
It doesn't necessarily grow back stronger, but it grows back, I mean, it calcifies, and we'll go through the repair, a simple repair of bone though. But when you break a bone, you have to create kind of this bony callus, and you're growing back, but it's never going to be, our body tries to make it as strong as it was. And so it sometimes overcompensates, because it's like when you dent your car and you put some putty on it to smooth it out. Sometimes you overdo it. So now you have more mass in that damaged area. And that's often what happens when you repair bones. You have a larger mass. So some people would say it's stronger because you have more stuff supporting it. But it's not necessarily stronger in the way that makes it better, because bones have to have a balance of being really strong and dense, but also flexible and bending like a tree. You know, so if a tree in the wind has a lot of give to it, it'll withstand a lot of forces on it. And new bone growth with that extra mass on there isn't always that resilient. So technically stronger because there's more mass, but functionally better, not necessarily. But it is solidifying in there, but there'll be other elements in there. But those would be osteoblasts that are taking place. Question?
I know we mentioned skeletal muscles. That's what actually happens when you tear down.
Well, if you're exercising, then that's hypertrophy. So, and it's not necessarily a tearing down, because there's a controversy between hypertrophy and hyperplasia. And so what you're saying implies hyperplasia, but it's not 100% proven in all cells, which is you don't tear down, because if you cause full-on damage and kill muscle cells, they don't always regenerate. So you want to keep your muscle cells healthy, but you want to put enough stress that tells your muscles, we need to have more mass. And so you don't want to damage, because then you're just repairing damage. You're backwards. So you want to put strain and force demand on your muscles that tells your cell, hey, we're going to need to have more equipment. If we're going to be like pulling this kind of weight, we need a little bit more stronger, you know, little tiny arms inside, which we will get to when we unit 2 about it. So there's a difference actually. If you have a myo is the prefix for muscle. A myocyte is a muscle cell. So we don't necessarily have myoblasts. So that's why I'm kind of making that distinction. But you have some muscle cells don't regenerate, and there's a very few that actually you can make more cells. We really are, the theories that are most commonly accepted is that we have a set number, and we just can make the same number bigger or smaller. And then there's a few instances where we can make more, but not very often. Function and location of different connective tissue types. So those slides where they're like, of those 3 things on each one, you want to think of big categories where we're going with the connective tissue. Bone and blood obviously is their own, but if we had the dense, and we had the loose, and we had the cartilage. So each one of those, so it seems like that's not too bad, but when you're like, oh, times it by 3, each one's got 3, it gets a little complicated pretty fast. So if we remember the dense, we had dense regular, we had the dense irregular, and we had the dense elastic. Then for the loose, we'll go here. Then we had our adipose. What else do we have for loose? Areolar. And then we had reticular. And for our cartilage, we had our hyaline. And we had our fibrocartilage. And we had our elastic. Okay. So I've got, and then on top, so all the connective tissues, and then bone and blood. So that's like everything for this whole category. So I'm going to make your life a teeny bit easier. Teeny, because I'm only going to take out the elastics for when we do the slides. So it's not going to be hugely easier, but this is where, so you should know the function and location of difference. So if you think of regular dense, who remembers what that was a part of? The strongest one? It's going to be filled with collagen, so it's going to be our tendons and ligaments. But then our irregular is also super strong, but in multiple directions, so it's going to be our dermis, not epidermis. Epidermis is epithelial tissue, so this is dermis. This is the next layer. And then elastic is just going to be, what was it, our, that one was this elastic. This one's vocal cords, dense elastic. So, but I don't usually, I have to remember because I don't usually do these. We usually do these, and then these first 2. So I don't make you learn visually the slides. We can skip the slide for elastic because some of the slides look like regular, and I don't want you to get confused by them. So I usually don't bother with it. But you should just know it's your vocal cord. For the loose, adipose, what is that? Fat. And areolar, what do we use that for? Yeah, just packing and just kind of support, but light support because it's not very strong. And then the reticular is the one unique one. It's a connective tissue, but where is it found? Spleen. It's like a filtering thing. And then hyaline cartilage is the most common cartilage. It's going to be ends of bones. It's going to be the ends of our ribs. You can see they're sort of representing the hyaline cartilage with this portion here. And we're also going to learn hyaline cartilage is going to be part in our growth plates. Fibrocartilage were the 2 places that you need to know about that. The menisci of your knee and intervertebral discs.